We will be talking on understanding the immunity of Newcastle disease in broiler chickens. And in between, if anything goes wrong, and if you're not able to see the screen, I would request the coordinator to put the flag up, right? So I make changes here. We'll be discussing on the understanding of immunity of against Newcastle disease in broiler chickens. And you can see I've written this, I have asked this question, is broiler immunity fragile against Newcastle disease? So we'll be debating on this point as well. And as we go through, I hopefully able to convey the message of the broiler immunity, whether it is fragile, or if it is fragile, what we do about it to reinforce the immunity by the end of the presentation. So the talk outline will be principles of immunity in broiler chickens. So I'll be talking on the basic arms of immunity that takes part in the protection against the Newcastle disease, or general outlay of the immunity in broiler chickens. Then correlates of protection, which arm of immunity plays to what extent, what the role in the protection of broiler chickens against Newcastle disease. Then we'll discuss on the susceptible period, or the immunity gap in the broiler chickens. And there we'll try to pinpoint the area where we need to focus to reinforce the broiler immunity against the or strengthen the broiler immunity against the Newcastle disease. And considering all these points, we'll look into an ideal vaccination program that can work out best in the field to protect the broiler chickens. So let's talk about now principles of immunity in broiler chickens. So basically, immunity or immune system could be, immunity conferred by the immune system could be of two types: passive immunity and active immunity. The passive immunity is conferred by the maternal-derived antibodies, which a chick acquires from its parent, while the active immunity is through vaccination or exposure or infection chick developed throughout its life. The active immunity, which is built through vaccination or field exposure, or if a bird gets infected, can be again divided into two types. Two arms. One is innate immunity, another is adaptive immunity. You can see I've written here memory. So adaptive immunity is something related to memory. So innate immunity, it has certain components inside, that is the phagocytes like neutrophil and macrophages, and the antigen presenting cells. What we see in innate immunity is the first line of defense. Whenever there is any challenge of antigen for the first time, or for any foreign object, maybe pathogens, so it first triggers inflammation, and once it comes into action, then it take it forwards towards adaptive immune response. So first of all, we need to understand an innate immune response is something non-specific. It doesn't recognize the pathogen or antigen, and it has no memory response. It is the first line of defense that is triggered through inflammation. After innate immune response is mounted, the adaptive immune response initiates, which creates a memory and prepares for the second exposure. So once the second exposure is there, the memory that is there due to the first exposure will help to mount more intensified immune response. And this is what is adaptive immune response. And it is very, very specific to particular antigen because it has a memory of these antigens or pathogens. So there are three components of adaptive immune response. One is mucosal immunity, which is correlated with secretory IgA, or local immunity, we also sometimes call it. But the most appropriate term to is terminology is the mucosal immune response. Then the cellular immune response, which is correlated with the T cells, or rather CD8 positive T cells. And finally, the humoral immune response, which is correlated with the B cells and producing IgM and IgY. So the memory immunity or adaptive immunity fades if there is no second exposure within few weeks interval. So that is why we need to boost, even there is an infection, the adaptive immunity is much more intensified. So if you're doing it, getting it done through a vaccination, probably, so the first exposure has to be boosted with the second exposure. Otherwise, the response fades away or the memory fades away. So this is what we have to keep in mind. Let's talk about the sites of this adaptive immune response. So where does the mucosal immunity play its role? Where does the cellular immunity plays its role? And where does the humoral immunity plays its role? Before I tell that, I would like to tell all of you, because I assume that all of you are veterinarians and we are qualified to understand. So I am not going into detail of these immune, these components of immune system. Otherwise, it's going to take winter webinar to complete it. So due to the paucity of time, I would like to restrict on some basic points only. So the mucosal immunity plays role in the tissue area, where it is correlated with the secretory IgA like tracheal washing, lacrimal fluid, and intestinal mucosa. The cellular immunity acts at the cellular level, like tracheal epithelial cells or enterocytes, whereas the humoral immunity, it acts in the blood circulation or the lymph. So when a mucosal immune response is taking place, it would be at the port of entry of the virus, because it acts at the mucosal surface of the organ. Like as you have seen that it acts in the tracheal epithelial cells or the lacrimal fluid, and it's the port of entry of the Newcastle disease virus. Intestinal tract or intestinal mucosa, it will act. So it is also a port of entry of a Newcastle disease virus when there is an enterotrophic challenge, that is through drinking water. If aerosol transmission is occurring through fomites or nasal secretion, then the port of entry would be the tears at the eye, the lacrimal fluid, and the trachea, where the tracheal washings bear the secretory IgA, which try to hold the virus at the site of entry. So mucosal immune response plays a role at this point of entry. Now, some basic points for humoral immunity or humoral immune response. So here I am to touch upon one more point that would explain the growth of immunity in broiler chickens. So what do you expect from a broiler chicken's immunity against Newcastle disease? Is class switching. Class switching is a phenomena where a class of immunoglobulin is changed its characteristics to another class of immunoglobulin, which is produced by the B cell. So whenever there is a first exposure of antigen, I could see through vaccination or field exposure, it will stimulate production of IgM type of immunoglobulins by the B cells. But later on, when there is a second immunization is taking place, or in other words, there is a second exposure of antigen, the immunoglobulin class, that is IgM, it gradually switches from IgM to IgY type. And here you must remember that IgY is significantly stronger in terms of virus neutralization or pathogen neutralization than IgM type. IgM is a basic immunoglobulin. It's a pentameric structure. The virus neutralization capacity is significantly poorer than the IgY type of immunoglobulins. So the point to be noted is that in broilers, you can expect that the first few weeks, it is mainly the IgM type of immunoglobulins, apart from the passive immunity, because in passive immunity, it is all MDA is always IgY type. But for active immunity, when you're giving it through when trying to triggering it through vaccination, the first few weeks has to be the IgM, which is going to protect the broilers. And it is quite weaker in terms of virus neutralization. We'll explain in detail when we go to the susceptibility or immunity gap, or the correlates of protection. Some key facts on the mucosal immune response: that the route of vaccination governs the site of mucosal immune response. For example, if you are giving an eye drop or nasal vaccination and to nasal route, it will elicit mucosal immune response in the respiratory tract. But if you're giving it through a drinking water, so it will elicit mucosal immune response or local immunity in the intestinal tract. Rarely, you are giving a vaccine that is growing in the respiratory tract or through intraocular route or intranasal route, it is not going to elicit mucosal immune response in the intestinal tract. So this is called compartmentalization of mucosal immunity, and also is governed by the live vaccine replication characteristics. Here, I want to mention one point. As I explained, that if you're giving the live vaccine to the respiratory tract route, like intraocular, that is eye drop route, or intranasal, it would elicit mucosal immune response in the lacrimal fluid and the tracheal epithelial cells in the form of secretory IgA. But it is not going to elicit mucosal immune response in the intestinal tract. But if a virus, which is much capable of replicating in the intestinal tract, and if you're giving it through drinking water, we have seen that it elicits mucosal immune response in the intestinal tract as well as in the respiratory tract. So the other way, it is not possible. So there are some theory related to compartmentalization of mucosal immunity, and also it is beyond the scope of this webinar to explain in detail. But for now, you please remember that compartmentalization of mucosal immunity says that the respiratory tract mucosal immune response is only related to application of live vaccine in the through respiratory route, either intraocular or intranasal. While in intestinal tract route, if you're giving it to drinking water, it will confer protection against the Newcastle disease by eliciting immune response in the intestinal tract as well as the respiratory tract. So through that route, you get protection in both the sides. But for here, the only condition is the virus has to replicate, effectively in the intestinal tract, because most of the Newcastle disease live viruses, they are, most of the lentogenic strains have poorer replication characteristics in the intestinal tract. There are certain viruses like vaccine viruses, like apathogenic enteric strain, like VH clone, VGYGA. If you're giving it through a drinking water route, it confers protection both in the intestinal tract as well as the respiratory tract in terms of mucosal immune response. I'll come to that also in detail when we discussed about just before the ideal vaccination program. But I don't want you to remember this concept before we continue to go forward. So class switching, already we discussed. This point was left for mucosal immune response, so I thought to go back and again talk about that. This is the cellular immune response at the cellular level, and it is correlated with the cytotoxic T cells and CD8 positive T cells. For details, we can talk about this again, in separate webinar, but it will be beyond scope to discuss here. One last point on the adaptive immune response is we must remember that the live vaccines that we use, it confers mostly the cellular and mucosal immunity and very less humoral immune response. While the inactivated vaccine, it has 100% humoral immune response, and they have no role in cellular and mucosal immune response. Any virus or vaccine virus that is replicating inside the chicken body will always confer cellular and mucosal immunity mostly. So live vaccines, and generally they have very less contribution to humoral immunity. To boost the humoral immunity, you need the inactivated vaccine. 100% humoral, they have no contribution in cellular or mucosal immunity. So with this background, I will try to explain you the correlates of protection for Newcastle disease virus. And hope you remember all those points. I touched upon some basic points, because without those basic points, the next section cannot be explained properly, or you won't be able to understand. So this takes us to a poll of the day. You see there are two questions. I would like to understand your thoughts. So I would launch a poll where two questions have been asked. Which arm is related to correlates of protection against Indian broilers? The first question is, which arm of immunity is having higher role in protection, do you think, against Newcastle disease, in order of its priority or in descending order? So you find the options are cellular immunity greater than the local immunity, than the humoral immunity, or the local immunity greater than cellular against the humoral. And you please vote your uh, consent. We like to see the results after it is completed. The second question is, which arm of immunity plays role in protection against ND in broilers in time dependent manner? Which plays the early role? Is it the MDA, then humoral, then cellular, then mucosal? You get the 4 options to choose from. I would request you, I'm getting only 4% vote. There are 382 attendees. I would request you to vote, as early as possible. Take your time, go through the options. It will help me understand, your thought process and your understanding and till now, and it will help you to understand, explain it better when we go to the next section. All right, we'll wait for more 30 seconds. I would encourage to complete the voting. We are 40% of the, out of 385, 160 votes has been casted. More 20 seconds to go. All right, so we end the voting here. We got 52% of the voting done, 206 out of 385. I'm sharing the result on the screen. So you can see 40% of you for the first questions think that the humoral immunity has the highest role in the protection. Then the cellular, then comes the local immunity. And for the question 2, majority, like 50% of you think that the first, the early protection, the maternal antibody plays the role. Then comes the local immunity, then comes the cellular, and last is the humoral. I'm impressed with the, with the results, because I think most of you have done in the right direction, and the rest of you who have voted for the other options, I'll explain you why it is wrong. All right, so let's move forward. So correlates of protection, the first thing is that the virus enters either to the intestinal tract route or to the nasal or intraocular route, and there the mucosal immunity plays the role in the respiratory tract and the GI tract. And it is circulated through different organs after its primary replication in the intestinal tract or the epithelium via erythrocytes. And this is called anemia. And in different tissue organs, the tropism is regulated by the fusion protein. And I'll be discussing in greater detail in the coming section. And this is governed by the F0 cleavage site, the host protease that will determine the virulence of the virus and the tissue tropism. And the agent characteristics as well as other protein contributes to the virulence as well. So what we're trying to do with the vaccine is we first want to generate the mucosal immunity at the site of entry or port of entry with CMI and secretory IgA production. And the CMI means cell-mediated immunity at the port of entry, as well as in the intestinal tract. Already explained about the compartmentalization of the mucosal immune response. If you're giving it to drinking water, we generate a lot of mucosal immune response in the intestinal tract. And if the virus is capable enough to replicate in more numbers in the intestinal tract, it depends on the vaccine characteristics, we get some mucosal immune response in the respiratory tract. But if you give in the respiratory tract, we get directly in the respiratory tract in fractal epithelium and the lacrimal fluid, the secretory IgA. And we do, sorry, I go to the previous slide again. And we prevent invasion of the virus to internal organs with the humoral immune response. That is the spread of the virus that you have seen in the previous slide from the first site of replication. And their humoral immunity plays the role when the virus is traveling to the blood circulation. So if we make a mapping of ND control, then the biosecurity will prevent the virus entry into the shed, or it will prevent the virus entry reaching towards the bird. The mucosal immunity will work at the port of entry, and the humoral immunity will work inside the here mucosal plus cellular both, and cellular also will target the tracheal epithelial cells inside the cellular level, and the humoral immunity will play the role in preventing the spread of the virus. Here also the cellular immunity will play to confer protection at the cellular level of each of these organs. So live vaccines is going to provide you mucosal immune response as well as the cellular immune response. Here mucosal means I'm talking about both cellular as well as the local humoral immune response, that is secretory IgA. And live and inactive active vaccines in combination will going to produce humoral immune response, profuse immune response. And without live and inactive in combination, you don't get antibody titers. So what should be our targets? The target is to prime the live vaccine, prime with live vaccine for mucosal and cellular immunity. And then you, boost with the inactivated vaccine. And our target titer for broilers is to achieve the 4-log2 in HI titer as early as possible. Here, layer and breeders are also mentioned. You can ignore that for now, but just to mention that it should have a titer laying more than 8-log2 or in IDXS ELISA at least greater than 14,000 IDXS. Why ELISA is not being preferred for broilers, I'll come to that when I explain about the immunity gap. You should focus on doing HI titer for broilers, which is more relevant, and it gives quite a real estimation of the protection level when you measure the titer, and I'll explain why. So priming with live vaccine and boosting with inactivated vaccine. Here we need to understand that why humoral immunity is so important. And I was explaining, I've asked this in the question in the poll, that which immunity has the highest correlates of protection, whether mucosal immunity, cellular immunity, and most of you have answered that humoral immunity has the highest correlate of protection. Here we'll understand why. So you see, this is a typical Newcastle disease virus, and you see two surface protein, the fusion protein, it's a mushroom shaped protein, and a Y-shaped protein that you see, it's a hemoglobin and neuromyelin surface protein. One is HN protein, another is fusion protein. Out of all the proteins, the surface protein, the fusion protein, HN protein is extremely important in viral infection. The HN protein, the Y-shaped protein that you see, please remember this structure, this Y-shape, and this is a mushroom shape. Because it's important, I have a graphics to explain you something. So the HN protein, this plays the role in the attachment to, suppose this is the chicle epithelial cell, and this is the Newcastle disease virus. This HN protein will bind with the tracheal epithelial cell sialic acid receptor. Once the HN protein binds the sialic acid receptor, the fusion protein on the virus will fuse the viral envelope with the chicken cell, lipid, phospholipid membrane. Once the envelope, the fusion process is completed, this virus will inject its viral RNA inside the chicken cell, and the replication process begins. And this is the stage where the infection process starts. That is the injection of the viral RNA inside the chicken cell. And there will be millions of copies produced, and this cell will be infected with the Newcastle disease virus. So the HN protein this helps in the already told in the attachment process. Without attachment, the fusion protein is not going to work. In this slide, I'm explaining this, that the HN protein, after binding to the salic acid receptor, will send signal to the fusion protein, which otherwise is not in structurally structurally it is inactive. It changes conformation to become active, and then the fusion protein takes place. So without HN protein, attaching with the salic acid receptor, the fusion protein morphology, the conformation doesn't change and it remains redundant. The signaling process is very, very important. And then only the infection process begins. So if you can block the HN protein at the very first step, the signaling will not occur, and the fusion protein, it remains redundant structurally, and the infection process stalls. There is no further progress of the infection. And this blocking can only be done by the immunoglobulins, even the secretory IgA, and when the virus is spreading in the blood, by the IgY and IgM. Better if it is IgY. Here comes a video that you can see. Here it will explain why the signaling process is important, why HN protein is so important, and why it is important to block the HN protein at the very first step. And what is blocking the HN protein is the immunoglobulins, and that is why humoral immunity is so important. If you have strong humoral immune response against Newcastle disease, you don't get anything in layers, breeders, and broilers. Broilers, it's sometimes tricky. I'll explain why, but in layers, breeders, we don't see any problem. So this structure that you see over here is a HN protein, and this mushroom structure is a fusion protein. And the thread-like structure that you see here is a salic acid receptor, and this is the chicken epithelial cell, and this is the virus. This is the HN protein. Here is the video for you. So the HN protein is binding with the salic acid receptor. This is tracheal epithelial cells of the chicken. You see the fusion protein is mushroom structure. In mushroom shaped structure that you see, it is structurally inactive. It is redundant. Once the HN protein binds with the salic acid receptor, it sends signals to the fusion protein, and it starts changing its morphology. The conformation changes, and this conformational change is only triggered by the binding of the HN protein with the salic acid receptor. Then it sends signal to the fusion protein, the right hand side, and then it changes structure. The structural change is important for fusion of the two viral envelope with the chicken membrane, and then only the viral RNA will enter the cell. So you understand now that HN protein is the most important protein for infection. If we block it, then only the infection process stops. This is a paper published in 2002, which shows that Newcastle disease virus HN protein alters the conformation of fusion protein at cell surface, and that is why HN protein, if you block, the fusion protein is vestigial. It has no role, and the signaling process stops. One myth I want to break here is that many marketing companies tell that you need to have genotype 4 vaccine, genotype 6 vaccine, genotype 7 vaccine, or you need to match the genotype of the circulating virus in the field with the vaccine virus that you are having. It's not true. That is why I've shared this concept with you. The genetic classification of Newcastle disease virus is based on fusion protein sequence and not on the HN protein sequence. And the good news is that HN gene sequence, or the gene coding for the HN protein or the HN protein sequence, doesn't vary across the NDV isolates. It's 0.1 to 10% variation, and which is fair enough. I mean, there is a homology of 90%, means you get a full protection. And it's conserved across the NDV isolates. The fusion protein amino acid sequence, of course, changes, and that is why, based on the fusion protein sequencing, the genotyping is done. And as I have told you, that the HN protein is the main protein in the infection process. And if you block with strong anti-HN IgY antibodies, the infection process cannot move forward. It stalls there. So, regardless of the fact that the field virus may vary based on its fusion protein sequence, having a conserved HN protein sequence, your genotype 2 vaccine, most of the vaccines are of genotype 2. So the genotype 2 vaccines you are using for live as well as killed is going to give solid immunity against the Newcastle disease, provided you maintain the titer or adjust the vaccination program to achieve that target titer of 4 log 2, or for layers and breeders greater than 8 log 2. Right. So the message here is that the selection and matching of field and vaccine strain is not required for protection against this Newcastle disease. The autogenous vaccines are velogenic in nature and they have very poor immune response against Newcastle disease. So rely on the genotype 2 vaccines out there in Nepal. That would help. So you understood the background of susceptibility correlates of protection now, and why humoral immunity is so important. In later part, I'll also explain why mucosal immunity and cellular immunity is important, because here I want to touch upon the highest correlate of protection, that is humoral immunity, and why it is so important. And this is why it is so important. And now we'll discuss on the susceptible period or immunity gap in broilers, or especially debate on the fact that how fragile is broiler immunity compared to layers and breeders. So this takes us to the second pole of today's talk, that broiler susceptibility to ND infection. According to you, I would like to know, what do you think is the most susceptible period for NDV incubation in broiler chickens? You have 5 options to choose from: day 1 to day 7, day 7 to day 14, day 14 to day 21, day 21 to day 28, day 28 to day 35. Let's see. I request you to vote, all of you. 391 attendees, please let me know. So 1 minute has passed. We'll wait for more 30 to 40 seconds. And we received 63% vote. 250 out of 387. Which is the most susceptible period for NDB incubation? I'm not talking about outbreak. I'm not talking about field case, that when you see the most outbreaks at what age. It is about the incubation period. Incubation period, if somebody who is non-vet over here, is the period when the virus is replicating inside. And it's the time from entry of the virus in the bird till the time the clinical symptoms show up. A period where the virus prepares itself, or it breaches the bird's immunity and prepare for the infection. That is called the incubation period. I assume most of you are veterinarians, so I didn't explain the incubation period. But if somebody is non-vet out there, so this is the definition of incubation period. All right, so we received around 73% vote, 287 out of 389. We'll end the poll and share the results. It seems that 45% of you think that it is day 7 to day 14 is the most susceptible period, while 20% of you, 26% of you think that day 14 to day 21 is the most susceptible period, and rest 20% thinks day 1 to day 7, and 3% of you thinks that day 28 to 85. So let's see which period is the most susceptible for broiler ND infection in broiler outbreak. So you can see this graph. The y-axis is the ND-HI titer. This is taken from the field cases that we see. The Zydus in India, we are the leading company who do this serosurveillance, massive serosurveillance we do, and we generate a lot of data in the field. Our technical team under me, they are working day in, day out, along with our sales team. Most of our sales team are trained to do this activity. So we do HI test as well as ELISA. In broiler, we prefer HI. So this is based on that data. So the maternal antibody generally varies from 6 to 8 log2 titer. So I've taken the base level that we see normally in the broilers, around 6 log2. It will drop to 0 by day 20. And this 4 log2, as I told, is the critical HI titer limit. And 2 log2 is the base limit below which you will see complete washout in the flock. So 2 is the minimum titer that we maintain. The critical titer is 4. And if you can achieve 6 log2, that's will definitely protect the broiler flock. So for now, we are considering 4 log2 as the critical HI titer limit. So a broiler chick starting with the MDA level of around 6 will have the titer drop down below 4 log2 by day 9 to day 10, and below 2 log2 by day 12 to day 14. Some of the birds which are 7 to 8 log2 will extend towards day 14. So from day 10, it falls below 4 log2 and continue. And this you see, this green line that you see is a humoral immune response, and this is due to vaccination. So I have taken a case where we are vaccinating with 3 live vaccine here on day 1, on day 9, on day 10, and on day 20 or day 21. Why day 9, day 10? I'm explaining you why, because most of you may not follow this vaccination program. But here, let's take an example here. If you do a vaccination on day 1 and day 9 or day 10 and day 21, what is going to happen? The active immunity will kick in gradually. Suppose you measure a titer around day 8, you won't see any active immunity. What you measure here is the declining maternal antibody. Most of we think that day 9, day 14, day 12, whatever we are measuring is the active immunity. No, it's not active immunity. It's the passive immunity. It's the declining maternal antibody, because it will be clear if you do another sampling at day 12, you will see it is coming down. Suppose it was around 3 log2 at day 8. If you measure at around day 12 or day 14, almost it will be close to 1 log2, or sometimes 0 if it is falling starting from 5 log2. So that will be clear. Then it will be clear to you that it's not the active immunity, it was the declining passive immunity that we are measuring. So literally a chick is susceptible to ND infection as the titer starts falling below 4 log2, and it is completely susceptible to mortality. I mean, when titer falls below 4 log2, the respiratory infection, the viremia, the chicks get more susceptible to all these things. But when the titer falls below 2 log2, absolutely at the first stage there is going to be mortality of the bird. I mean, the incubation period will be much lesser for the ND. So the 2 log2, in my opinion, is the minimum or bare titer limit that we have to maintain. So by day 14 to day 20 or 21, you see the maximum of the chicks are below 2 log2, because by that time the active immunity is not kicking in and the passive immunity has fallen below 4 log2 and further below 2 log2 by day 14 to day 20. So the first ND live, what role it's playing? The first ND live vaccine, once you give it, it will stimulate the secretory IgA if you give it in the intraocular route or intranasal route, it will stimulate the secretory IgA. It will stimulate the cellular immunity, but it will be neutralized by the MDA. I mean, so the first replication in the trachea would stimulate the cellular and secretory IgA, but as soon as it goes inside the blood circulation, it is going to get neutralized by this massive MDA. So once it gets neutralized by the MDA, the humoral immune response elicited by the first live vaccine is negligible. So you can't expect any humoral immune response with this ND live. And I told you also on this one more point at the beginning that live vaccines are responsible more into cellular and local immune response or mucosal immune response. It has very less contribution in the humoral immune response. But with this live vaccine, you get nothing because it gets neutralized by the MDA. What happens with this second ND live and why we are giving at day 9 or day 10, most of you have understood. We are waiting for the MDA level to drop down. If you give at around day 4 or day 5, it makes no sense. Again, it's going to get neutralized. Or if you give the first vaccine on day 5, it again makes no sense because the role of the first vaccine will be to stimulate the IgA and cellular immunity as early as possible at the port of entry. So there's no same, it makes no sense that you delay it from day 1 to day 5. If you give on day 5 or day 1, in both the cases, it is going to be neutralized by the MDA. So the humoral immune response is not going to happen. We need to get the mucosal and cellular immunity as early as possible. So it has to be given on day 1. For the second live, its role is again in the mucosal and cellular, but to some extent to humoral immunity, because by that time the MDA level drops considerably, close to 2 or lesser than 2 log 2. So this vaccine we have seen given, it kicks the triggers the humoral immune response. And after that, when we give the next vaccine on day 2021, it further takes it forward. So it plays again role in mucosal and cellular immune response and again humoral immunity. But the window of susceptibility will always remain because the gap between the MDA dropping below 4 log 2 and further below 2 log 2 and the time taken by the active immunity to rise up to 4 log 2 or at least above 2 log 2 will be there with the live vaccines in place. So again, there is a challenges of class switching. And that's explained why there is a fragility of immunity in broiler chickens, even if you achieve 4 log 2. See, the MDA is of IgY type. So till the MDA is above 4 log 2, absolutely your flock is fine. Till day 10, you don't see any incubation happening. For that, you need to have a good maternal antibody. Most of the breeders are well vaccinated. So the maternal antibodies would be close to 6 log 2. In some cases, we see even 7, 8 log 2, 9 log 2, even maternal antibodies. So these are of IgY type. They play a very crucial role in preventing early challenges. And on top of that, we have already given an ND live on day 1. So that will also add to the mucosal immune response and cellular immune response from day 1 itself. Now, when you give the second live, it has some role in the humoral immunity, as I told, because the first one has no role. And first few days, if you give, it's going to be neutralized. So the first immunoglobulin that comes into the circulation is IgM. Here is the challenge of class switching, and it is very weaker in terms of virus neutralization. And this class switching happens slowly based on the exposure. After this vaccine, we got a field exposure, and that field exposure, as well as if you are giving the second vaccine, slowly the class switching is happening. So until day 21 or day 25, you don't see any IgY immunoglobulins. And that is the reason if you do ELISA, you can't get any titers. You get very bare base titer. Even in HI, you can see 4 log 2. But if you suppose you are doing ELISA at day 24, the same sample you are doing HI titer, in ELISA, you won't see good titers. The values will be coming very close to the the negative control or the blank. It is because the ELISA that we use here measures anti-IgY, ND-IgY molecule. The trapping antibody, the conjugate used in the ELISA is IgY type of conjugate and in indirect ELISA. So it measures the IgY antibodies only. But here we need to measure even the IgM and IgY type. So that can be that HI test has a sensitivity towards that. Then so it's better to do HI test in ND against ND in broilers, because in first few days you get IgM and slowly class switches to IgY. So if you see the arm of immunity, the mucosal immunity plays an important role in the from day one itself, and keep on vaccinating with live vaccine. It will continue to do that. The cellular immunity will start at least 3 days it would take to trigger after you have done the live vaccine on day one. And the more we do live vaccine, it is in action. But the most strong immunity, arm of immunity, that's the humoral immunity, it comes very late. And once it comes with the IgM type. That is why the broiler immunity is so fragile compared to the laying and breeder birds, because the most stressful period of the laying hens is during once it starts laying. And by that time, it achieves good amount of immunoglobulin produced in the blood circulation, the cellular immunity, and all are of IgY types. That is why it can provide solid resistance to the kill challenge. But in case of broilers, we have this window of susceptibility. We have the humoral immunity, the strongest arm of the immunity against ND, coming late in the picture. And although it comes late in the picture, on top of that, it is the IgM type that that is predominantly there in first 3-4 weeks of their broiler life, which slowly class switches to IgY. Now, this is a scenario. If we combine a ND live with ND inactivated vaccine, what will happen? This window period will at least close down by 4 to 5 days. And this is enough to reduce a lot of mortality, respiratory challenges, respiratory diseases. And even there is no outbreak, broilers undergo respiratory distress. If the titer is between 4 to 2 log 2, you will see respiratory distress. If titer comes below 2 log 2, the challenge further progresses and ends up in death of the bird. So you can reduce the respiratory distress considerably, and you have lesser window of susceptibility. And so consider using a vaccine or an inactive vaccine on day one. But this should be the program. If you are doing a vaccine on day 4, day 5, it's going to be neutralized, and not doing any vaccine on day 9, day 10. There is nothing there that can kick in the humoral immunity. So it has to be there. Already explained what is the role of each of these vaccines at different stage. So the take-home message is live vaccines alone create large window of susceptibility, can be reduced by high potency of good quality inactivated vaccines. Class switching is slow with live vaccines. It can be expedited by high potency or good quality inactivated vaccine. But remember, the 3 live vaccines has to be given as mentioned there. I'll come to that again later on, because without them, you cannot achieve the humoral immune response. So this part already explained in the previous slide. Just a reminder that you need to have live vaccine given through both the routes. Initially, eye drop route, which is going to be neutralized at the blood circulation level, but it will stimulate the secretory IgA and cellular immunity. While the later part of the broiler live grow out, you have to give the second or third live in the drinking water so that it elicits a mucosal immune response in the intestinal tract as well. So you get a mucosal immune response and cellular immunity at both the sites. Even the systemic cellular immunity will also be more if you are covering it up from both the sites, intestinal tract route as well as the respiratory tract route, and combined with the inactivated vaccine for better class switching, faster class switching, as well as to reduce that window of susceptibility by 4 to 5 days. But it's still there. That is why the broiler immunity is fragile. We need to help it with the biosecurity to reduce the viral challenge as low as possible. Here, just I'll briefly touch upon why mucosal immunity is so important here after the humoral immunity. I just take an example of a vaccine of our company. I know it's not there in Nepal, but it is very good example to take. That VH clone is a pathogenic enteric strain. It is developed by 2-step cloning process. So it can replicate both in the respiratory site as well as the enterocytes. And it is a clone vaccine, so the immune response is uniform. So here is a classical study done that you give a Lasota and B1 vaccine in the respiratory tract. This is the level of secretory IgA produced, the mucosal immune response in the respiratory tract. This is 3 days after vaccination. This is 7 days after vaccination. But when you give a Lasota through drinking water route, the production of IgA is extremely, extremely poor. In fact, there are several other papers which has confirmed with this study that the Lasota strain, being a lentogenic type of pathotype, it varies in its efficacy in terms of replication in the enterocytes, strain to strain. So this is again the compartmentalization theory of mucosal immunity, where if you are giving a vaccine which is not able to replicate in the intestinal tract, neither you will get a mucosal immune response in the intestinal tract, also you won't get in the respiratory tract. So Lasota and B1, I would recommend if you're giving, only restrict to the respiratory tract, but not in the early days of chicks, because it causes lot of stress. And again, B1, although it replicates, it gets easily neutralized by MDA. Try to have a clone vaccine, whatever there is available in Nepal. This is one more study with the VH clone. Being an apathogenic enteric strain, it was given on day 1 through intestinal tract route and respiratory tract route. What we see here is detection of the antivirus through RT-PCR at different tissue sites on trachea, lungs, duodenum, and cecal tonsil. So day 1 after application, you find a duodenum. When you give VH clone in the drinking water route, you find in the after day 4 in the the tracheal epithelia, the lungs, the duodenum, cecal tonsil. It continues to be there in the trachea, lungs, duodenum, and cecal tonsil, and it disappears from the trachea and lungs and continues to remain in the duodenum and cecal tonsil till day 11. But when you give a Lasota strain through the intraocular route or respiratory tract route, it appears only in trachea and lungs. It doesn't come in the duodenum and cecal tonsil. Even there is no question of giving in the drinking water route, because the replication characteristic of Lasota is very poor. So that also proves the compartmentalization theory of the mucosal immunity. The only condition is that the virus has to be replicate in the enterocytes. So this is a kill vaccine. In kill vaccine, the criteria would be the antigen concentration as well as the adjuvant quality. Nowadays, vaccines are registered on the basis of PD50. So to make you understand about the PD50, how to get a strong humoral immunity and how to judge a vaccine available in your country is to see the PD50 and EID50 values rather than EID50 values. For the live vaccine, already mentioned, to see if it is cloned or not, the ICPI index has to be lesser, as well as it needs to replicate both in the respiratory and the enterocytes. For live vaccine, this is the most important criteria. For kill vaccine, look for the antigen concentration and then ask for the PD50 value of the inactivated vaccine. EID50 is not the correlate of protection, in today's world. The EID50 measures the antigen concentration, whereas the PD50 measures the actual protective dose. So this is the criteria of protection for Newcastle disease right now. The PD50 value that is recommended is the minimum volume that should have a value greater than 35 for protection against Newcastle disease. So try to have a vaccine that has a value close to 100. The PD50 value should be close to 100. Don't look into the EID50. PD50 gives the actual correlates of protection now. Earlier, people used to see the EID50 or antigen concentration. So for example, if you see in European pharmacopoeia, the PD50 measure was adopted, and from last 3-4 years, they are following the PD50. And India also, we are looking into PD50 values for judging the efficacy of the inactivated vaccine against Newcastle disease. In European pharmacopoeia, it should be greater than 35. I think they have now made it, it should not be less than 50, and it should be greater than 35. So, for example, Nectifort, we have the inactivated vaccine. It has a PD50 value 100, 0.1 for 0.1 ml dose and 0.2 ml dose 200. So this is how we judge. And say competitor one comes around 50 compared to 2 comes around 116 to 126 for 0.2 ml. So this is how we judge the PD50 value, I mean, the efficacy of the inactivated vaccine with PD50. This takes us to our, the next or last, outline of talk, that is the ideal vaccination program. So with all this knowledge, I hope you are able to understood that what should be the ideal vaccination program for Newcastle disease. It takes to a third poll, the same question: which program do you follow, or you would like to follow after this discussion for Nepal? Your voting starts now. One minute has passed. We have received 41% vote. I would encourage to go ahead and see which you think as the ideal vaccination program among the 6 options to choose from. All right, more 10 seconds to go. All right, we end the poll here. So we have received 229 votes, and here is the result for you. So majority of you think the ND live and ND inactivated vaccine on day 1, day 9 ND live, and day 9 ND live to be the ideal. And whoever, I think there are 22% of the voted attendees thinks that day 1 ND inactivated vaccine, day 5 ND live, and day 9 ND live to be ideal. I would like to show this, keep this screen shared on. I explained to you that 5-day ND live, delaying it to 5th day makes no sense, because if you are delaying it, eventually gets neutralized by MDA. We need to understand what is the goal of this vaccine, or what this vaccine is capable of doing. The live vaccine on day 1 to day 7, they are going to be neutralized by maternal derived antibodies inevitably. We can expect stimulation of cellular and local mucosal immunity from the live vaccines during the first week. So why we should delay it? Delaying that vaccine makes no sense, because that is the first line of defense. You see, we understand we need to get the humoral immunity as fast as possible, but there is no other way to do that. Broilers has a shorter life cycle. Its immune system is naive. The immune stimulation is not as faster as an adult bird. On top of that, it is the bird having highest stress among all classes of poultry. Considering layer breeder, the first 35 days, the body weight achieved is not even close to the bird, a layer bird in their entire life cycle. A layer bird stress start from around 17-18 week, when they start just laying eggs, the reproductive organ development from 14 week onwards. But for a broilers, from day 1 itself, there is a mammoth rat race, and they achieve the body weight gain they have. Even the breeder and layer, they even don't come near. In my calculation, they have 10 times more stress than these birds at that period, that age, from day 1 to day 55, compare a layer, breeder, and broiler. So I do delay the first live vaccine. There's no point of delaying that. Day 9 vaccine is required, absolutely, because without that, this ND kill vaccine that you are doing on day 1 will not flare up. I explained a kill vaccine triggers humoral immune response. It cannot without a live vaccine. It is called prime boost immunization strategy. In my previous slide on the correlates of protection, you could see that live and inactivated vaccine combination can produce AB plus. I written there because that is called prime boost immunization strategy. So you need to have a live and killed in combination. So let's go to the next slide. And so here it is. So this is a vaccination program I would like to recommend for you. So this is our actually we have a IBD vaccine that can be given in the hatchery. This is called MB1. For now, you just consider NDVH. I'm writing it's a ND live, NDVH and IBH 120 live. If you're doing in a very high challenge area, combined with a ND killed vaccine, and then with ND and IB live, on day 9 or day 10, you do ND live. This hashtag you see is because day 9 vaccine should be postponed to day 10 if chicks ND MDA level is greater than 4,000 IDXs or greater than 6 log 2 HI titer. The goal is to achieve greater than 3 log 2 during 15 day to 18 day of age, and that is the most susceptible period, that is day 14 to day 21 period. So I asked this question, the most susceptible period is between day 14 to day 21 years to achieve faster humoral immunity, at least above 2 log 2, touching 3 log 2 during this period. So that is why this day 9 vaccine is critical. Day 9 vaccine will ensure because it is least neutralized by the maternal derived antibodies, and more it would be playing very crucial role in the systemic cellular immunity as well as the humoral immune response. And this will actually, day 9 vaccine will actually help the ND kill vaccine that you have done that only contributes to humoral immune response to work very well, to flare. This flare won't be occurring without a middle vaccine in place, that is A9 vaccine. So somebody will be doing day 1 ND kill, day 5 ND live, and day 19 ND live will not have proper immune response between day 14 to day 20. You will have an immune response after day 20, 21, 22, 23, humoral immune response. And it makes no sense. Again, I found some people doing the serosurveillance at day 35, HI test at day 35. And HI test is one of the ideal method of serology for the broilers. It makes no sense. If you really want to do a serology, do on day 18 to day 20 or day 15 to day 20 and till day 24. If you can protect the bird, achieving the titer between day 15 and day 24 as fast as possible, and if you're judging the efficacy of vaccines available to choose from among different competition in Nepal, do it between day 15 to day 25. Doing titer tests at day 28, day 35 is less significant. You can always do that, but it's less significant than this doing titer test or doing serology between day 15 to day 25, because that is the most susceptible period, day 14 to day 21. I hope you have understood the main concept of the ND immunity. And with this note, I want to thank you all for joining us. And I want to thank Vetnepal